Hello, good afternoon, everyone. It's already 13:48. So can we start for the new session in this room, Biotechnology B? Can we start? In this new session, we will have another 4 presenters. They are Ibu Maulidia, Ibu Linda Sukmarini, Bapak Syah Putra Wibowo, dan Ibu Tati Kusniati. So those are our presenter for this session. So the first presentation will be delivered by Ibu Maulidia Rahmawati. She will talk about silver nanoparticle elicite culture, a nanotechnological approach of secondary metabolite enhancement for plant improvement and medicinal important substances production. Ibu Maulidia, are you there?
Yes. Can
Okay.
you hear my voice?
Yes, I can hear your voice.
Okay. Can I share my screen?
Share. Please do share your screen. Since you already be a co-host in this meeting.
Okay. Is it shoot? Okay.
Yes, it's already on my screen.
Thank you. Can I start?
Sure, you can. Time is yours.
Yeah. Thank you for the nice opportunity. Good afternoon, ladies and gentlemen. Uh, Thank you for joining the session in this afternoon. I am Olidia Rahmawati on behalf of my team. Dr. Nurul Jadid would like to present our review entitled Silver Nanoparticle Elicited Culture, a nanotechnological approach for crop improvement and secondary metabolites production. As we know, that plant playing a very big role in fulfilling our needs as a human, from our food to our medicine. But several obstacles are found when the need of plant is actually keep improving. So plant tissue culture is basically one of the methods that most successfully solve this problem. And several modification of this method has been presented in the in the research before. So to break down the achievement of this method in solving the problem, I would like to discuss about the plant tissue culture and silver nanoparticle in general, its achievement in crop improvement and secondary metabolites enhancement, and also the mechanism of action from the silver nanoparticle in the elicitation of culture. So in vitro culture of plant belongs to non-genetic modified biotechnology. It works based on the totipotency theory and have to work in the aseptic condition with the controlled environment. It has been used widely in the basic bioscience, crop improvement, secondary metabolite production with diverse application and techniques has been presented. Living in the era of nano, all of our life has been in touch with nanomaterials and also nanoparticle. Nanoparticle itself defined as three-dimensional nanomaterials that have size from 1 to 100 nanometer with different types and shapes. This particle has a unique characteristics such as low weight, large surface area to volume ratio, electron exchange, and surface reactive. Silver nanoparticle itself has been gaining more interest worldwide because of its potential as antimicrobial as well as its positive impact in the plant cell growth, biomass accumulation, and phytochemical production. This nanoparticle, known as having its hermetic effect, that is described as having uh, supporting impact to the growth in the low or medium doses and also inhibiting it in the higher dose. The research from Xu et al. In 2014 have shown the different shapes that could be produced from the silver nanoparticles. Several studies has exhibited the potential of silver nanoparticle in improving plant growth in diverse plant species. This, this showed by the evidence of it by providing contaminant-free culture medium, effect on explant multiplication, and also biomass improvement. Research from Abdi et al. Showed that the application of silver nanoparticle after surface sterilization inhibit the Xanthomonas sp without adverse effect on the plant growth that been observed. It's also been supported by Li et al. That showed that silver nanoparticle in the size of 3 nanometer successfully control pathogen fungal strains. The further research from Sarmas and Salehi in 2016 also showed that media supplemented with 18 nanometer silver nanoparticle with the concentration of 400 microgram per milliliter postponed internal contamination in A. excelsa to 18% compared to the control, which have the number of 81%. Silver nanoparticle elicitation has also been reported having a good impact in the callus proliferation and plant multiplication. In Prunella vulgaris, known as important medicinal plant, known as self-hull, 100% of callus proliferation was observed in the treatment of 3 microgram per liter of this nanoparticle. This also showed the enhance of biomass accumulation after 30 days of period. In sugarcane and stevia, the addition of uh, low and medium dose of silver nanoparticle with the size about 35 nanometer significantly increase shoot multiplication and elongation, while uh, the higher dose of it exhibit the decrease of the growth of the plant. And then in Oryza sativa cultivar ER64, 10 milligram per liter of silver nanoparticle reported to increase 20% of callus induction. And its combination with hormone as nanobullets or known as hormone stabilized nanoparticles, showing that this method increased rooting result in the Nicotiana tobacco. In the callus biomass, the elicitation of 60 microgram per liter of silver nanoparticle along with 2,4-D and also BA presented 60-fold increase of callus biomass in Caralluma tuberculata compared to the control. It shows its promising result for this endangered species ex situ conservation. In chrysanthemum, elicitation of 1.5 ppm silver nanoparticle in both liquid and solid medium increased shoot, plant height, and also biomass. Silver nanoparticle also reported to significantly enhance the production of plant secondary metabolites and antioxidant-related products to the observed plants. In Caralluma tuberculata, elicitation of 90 microgram per liter of silver nanoparticle known to increase the TPC or total phenolic compound, total flavonoid compound, PAL, and DPPH free radical activity. Well, in sugarcane, 50 50 milligram per liter of silver nanoparticle significantly increased the phenolic compound production. While in the while in the model plan of Arabidopsis thaliana, 10 nanometer of silver nanoparticle in 5 ppm known to significantly increasing gamma-lexin synthesis compared to other treatment.
Pardon,
For
Ibu Maulidia, you only have 1 minute left, so make use it well. Okay.
for the cell suspension culture, it has been done in the chili and the bitter gourd, knowing that this addition of addition of silver nanoparticle could increase its capsaicin content and also the TFC and TPC in the bitter gourd, with increasing also if in antimicrobial and pharmacological activity. The possible mechanism of silver nanoparticle has been established the re- for the initial response of the plant to the silver nanoparticle. It is known for the ROS bars that enhance the transcriptional programming that affect uh, the trans the effect of this to the PGR related genes that could increase the yield of the plant, such as in abscisic acid and the ethylene that the silver nanoparticle elicitation known to have the decreased impact of the culture. From the breakdown that we have been discussed before, we can take the conclusion as that elicitation of silver nanoparticle in plant tissue culture revealed improvement in growth and secondary metabolites production. Silver nanoparticle elicitation shows hormetic effect in tissue culture. This depends on plant species, the size and the concentration of the silver nanoparticle. And silver nanoparticle reacts with plant cell via initial ROS activation. Silver nanoparticle elicitation on plant tissue culture is a promising way for large-scale crop improvement and secondary metabolites production. That's all from me. Thank you for your kind attention. See you.
All right. Thank you, Ibu Maulidia. Enchanting presentation. So we move to the second presenter. That will be Ibu Linda Sukmarini.
Kamu sadar nggak sih kameramu tuh mati sepanjang mati?
Iya.
Halo Bu Linda.
Yeah. Okay, I'm here. Can you hear me?
Share I can, Ibu.
May I share my screen?
Yes, please do share your screen to us. All right, Bu Linda, time is yours. You can start your presentation by now.
Okay, good afternoon everyone. Assalamualaikum
Good afternoon.
warahmatullahi wabarakatuh. I'm Linda Sukmarini from Center for Biotechnology. First of all, I would like to thank the IBOC committee for having me this opportunity. Today, I'm going to present a review about recent advances in discovery of lead structures from natural products. Here are the outline of my talk today. It's mainly covering bioinformatics derived approach and also bio. As a background, natural products are secondary metabolites originating from plants or from animals, such as from the phenom. from marine organisms and symbionts, and notably also from microbial sources. They inspire many discoveries in chemistry, in cell biology, as well also in medicine or drugs. Natural products possess a vast structural and chemical diversity with a broad spectrum of biological properties or biological activities such as antibacterial, antifungal, cytotoxic, anti-tumor, or anti-cancer. And, and interestingly, more than half of the new small molecule drugs were have been developed so far from microbial natural products in over last decade. So here my focus of my talk will be microbial natural products. The current interest in the discovery of natural products, in particular microbial natural products, derives largely due to the failure of synthetic libraries generate an expected number of drug candidates in pharmaceutical industry. And in addition, also the clinical relevant pathogen resistant or antimicrobial resistance have uh, warrants the search for novel bioactive metabolites from the microbial natural products. However, searching novel or new natural products from microbial natural products, it's become more difficult because the high rate rediscovery of non-natural product by limited classical or conventional methods such as bioactivity screening and chemical signature guided isolation. So therefore, here today, I try to give some example in technological advancements in order to accelerate the discovery of microbial natural products. One of well, one of these example is metagenomics approach. Metagenomics simply relying on sampling of environmental DNA that we call also eDNA, usually from organism or strains that usually difficult to isolate or to cultivate. The revolution in metagenomics have been so far uplifted by the advancement in the next generation of sequencing technology. And one of the example of the application of metagenomics is the discovery of malacidin by capturing this DNA from the soils and isolate and also identify their BGC, and then we try to express them in heterologous host in order to lead a new natural product, malacidin. And another example of this approach also the discovery of bifamides. Bifamide is an antiviral peptides against HIV virus, which synthesized from the uncultivated microbial symbionts of marine tunicates. Another example of culture independent technology is the recent we call it iChip or isolation chip screening method. It's very simple chip compartment, and so far it has been successful to discover teixobactin. Teixobactin is a new antibiotic that kills pathogen without detectable resistance. We can say that in this genomic era, novel natural or new or novel natural product can be discovered by mining their genomic information. As you can see, as of May 2019, the NCBI, the public database, exists with more than 211,000 bacteria, which provide very rich genomic diversity, which is the potential sources of novel or new nature products. And here, the genome with containing the BGC linked to the structure of nature product. This the example of the genome of the Streptomyces avermitilis. Uh, 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 by bioinformatics study, it can reveal that within a single bacterial genome, 23 of the total predicted 40 BGC are silent under exploit culture condition. So that's for it's very promising for discovering novel natural product, and therefore the BGC or biosynthetic gene cluster must be prioritized by product novelty and or their function as well. So here are some of bioinformatics guided of genome mining tools to simplify target the BGC identification towards the knowledge of the biosynthetic machinery of the well-studied natural products such as for ribosomal peptide, non-ribosomal peptide, or for PKS. For example, you can use anti-smash for identify the NRPS peptide, or you can use RipMiner or Rodeo for detect the ribosomal peptide. So here are the overview of genome mining approaches and the staff involved, which is also correlate with other approach such as metabolomics, genome mining, in order, and also the computational analysis is mean like bioinformatics, which is need bioinformatics guided in order to obtain the new or novel natural product. And here we can see that two new NRPS PKS from June non-mycin A or West non-mycin A1 were discovered through BGC genome mining of promising anti-cancer lead from linemycin NP family. And then another example is thiotronic acid antibiotic, which was discovered by a resistant directed genome mining strategy. And another innovative strategy is the advancement in mass spectrometry-based bioanalytics. Mass spectrometry is a predominant analytical technique with wide applicability in high throughput screening programs, and recent advances in MS that integrate this molecular networking not only can replicate the known compound, but also identify the analogs of non-compounds, and also can contribute to unravel new compound by avoiding the re-isolation of the non-compound. And recently, the molecular networking has been coupled with biosynthetic gene cluster with the genome mining to dig more into the BGC responsible for the production of a metabolite. So MS guided genome mining, that we call it also metabolomic, can detect new natural product and connect them with their biosynthetic gene cluster. This metabologenomics works by grouping similar BGC into gene cluster family from diverse bacteria and lead to the the production of new natural product, such as, for example, this we have tambromycin using this application. Moreover, this metabologenomic also recently has been connected to the MSMS networking. And
Pardon,
to
Ibu
sum
Linda,
up,
you only have 1 minute
to
left.
sum
Oh,
up,
okay.
I would say that advancements in genomics and as well as metabolomics can accelerate the drug discovery mainly in microbial natural products. And with this all advancement and maybe together with the synthetic biology advance, it's promising that natural product research keep continuing to develop and many drugs will be discovered. Okay, thank you for your attention and I welcome for the question. Thank you.
Okay, thank you, Ibu Linda, for giving us a very nice presentation. Now we would like to move to the third presentation that will be delivered by Bapak Syaputra Wibowo. Bapak Syaputra Wibowo will deliver presentation about the density function theory study of astaxanthin metal complex to native and glycated human serum albumin. Halo Bapak.
Okay, thank you. Yes,
You're
thank
welcome.
you for the opportunity. I would like to present our paper. First of all, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Uh, First, we would like to thank the IBOC 2020 committee for let us to present our paper with entitled The Density Functional Theory Study of Astaxanthin with Metal Complex to Native and Glycated Human Serum Albumin. Our team consists of first me, myself, Syaputra Ibowo, and Dr. Sriwidiarti, Dr. Ahmad Sabarudin, Professor Dr. Joko Wahano Sutmaji, and the last is Professor Sutiman Bambang Sumitro. And let's jump into the introduction. As we know that diabetes mellitus is a degenerative disease that tends to experience complication due to the change of physicochemical of blood. This main characteristic of the changes in physicochemical of blood is a change in the red blood viscosity, which is very related to the functional aspect of albumin as a transporter and as a scavenger. In this diabetes mellitus case, is likely to increase free radicals. And as we know that free radicals are also the normal component in our cellular metabolism, but if the amount of free radicals are more than normal, of course, it can cause cell damage that impacts oxidative as well as molecular and is a major factor causes the emergence of pathological condition. And here we try to see the new perspective to maintaining harmony of this diabetes mellitus case, which using astaxanthin, while astaxanthin is a red cartonate pigment which doesn't have the activity of provitamin A and a biological antioxidant that occurs naturally in many types of plants, algae, and seafood. So, in order to seek this perspective to maintaining harmony, of course, we are doing the DFT and molecular dynamic. In DFT, we can see the electron density of any system determines all ground state properties of the system. And in this case, the total ground state energy of many electron system is a function of the density. Meanwhile, the molecular dynamic can help to understand the stabilization of protein molecule in a specific environment. And of course, the objective of this research are to characterize antiradical property of astaxanthin with CO2 plus and astaxanthin with 2 CO2 plus with density functional theory and to compare the capability to prevent conformational changes on glycated albumin. And before we are jump into the methods, I would like to say that our previous study showed that astaxanthin can be used as a therapeutic agent to preventing conformational changes on glycated albumin, and we already published the result in one of Scopus journal. And as astaxanthin as a single compound, and here we would like to show that astaxanthin could be complex with the metal ions, so it could improve the capability of astaxanthin to prevent glycation and product in albumin, human serum albumin. So in the methods, here data mining, Avogadro, Gaussian O9W, GaussView O6.0, docking with Pyrex, PyMol, Discovery Studio 2016, and the last is molecular dynamic using Yasara. And here we are going to the methods. First, data mining. As we know, the astaxanthin, CO2 plus, glucose, and metformin, we got it from the PubChem database. And albumin, we got it from the PDB or protein data bank. We use the HSA or human serum albumin with 4K2C. There is no ligand at that protein. And after that, we are input the isomeric smiles of the astaxanthin, and we are going to build the complex with Avogadro. And in the complex itself, we are using the astaxanthin with two types of complex. First is astaxanthin with one metal ions in the beta ion rings, and the second is astaxanthin with two metal ions in both of beta ion rings. And we are going to set a force field, number of steps with 500, and algorithm with stable design, and the last is optimized geometry. And the last is we are using the Gaussian O9W, and here we are talking about the density functional theory as implemented in Gaussian O9W was used for all calculation. In job type, we were choosing optimization and frequency with ground state and DFT with global hybrid three parameters or B3LYP, density functional, and the LAN L2DZ basis set. We choose this basis set because it works wonderful with organometallic compound. We were able to verify optimize minima using harmonic frequency analysis, and as previously reported, the density functional approximation calculation using LAN L2DZ pseudo potential are adequate for metal interaction, for example, those of zinc, cadmium, and mercury. And the absorption spectra of astaxanthin with metal cationic complex have been computed with time-dependent density functional theory, or TDDFT, using the same functional and basis set. And for solvation, we were using a polarizable continuum model with specifically the integral equation formalism, or IEF PCM, with ethanol as solvent in order to make a comparison with real-life experiment. And here we can see that after that, we are using the docking with Pyrex, Pymol, and Discovery Studio 2016 with the Venice search space as shown in the presentation. And the last is using the molecular dynamic with Yasara. We are going to use MD-Run, MD Analyze, and the last is MD Analyzers. For the result in discussion, there are three parts. First is the optimized structure, vibration, ER spectra, and the last is HOMO and LUMO. What is HOMO and LUMO? In HOMO is the highest occupied molecular orbital, which means that electrons from the HOMO are donated and also most available for bonding, most weakly held electrons, and the last is characteristic for nucleophilic component. And in LUMO is the lowest unoccupied molecular orbital, which is a received electron, lowest energy orbital available, and characteristic for electrophilic component. And the second part is electronic energy, energy in thermal, head capacity, entropy, dipole moment, and polarizability. And the last is binding affinity, potential energy, binding energy, and the last is movie visualization. And here we can see in optimized structure. In optimized structure, we can see the astaxanthin in the native form, and the vibration. There are two parts of vibration here in the left and right of beta ion rings, and in AR spectra we can see in 500 until until 0. We can see in the fingerprint area there are no value. No, there's no highest value. It shows that there's still no bounding in metal ions into astaxanthin compound. Of course, because astaxanthin is still in the native state. And here in HOMO and LUMO, we can see the pattern. And when we are adding CO2 plus in one metal ion, here we can see in CO2 plus in the right side of beta ion side, and the vibration become more different than the other. In this state, astaxanthin with metal ions, of course, will show that the vibration state is far more likely to become more general than before. And in ER spectra, in 500 until 0, you can see the fingerprint area show that the metal ions already bound into the astaxanthin. And in HOMO and LUMO, show the different pattern than before. So what does that mean? We can show it in the molecular dynamic and in the last section. And astaxanthin with two CO2 plus and two metal ion section in the right and left image here, there's a bonding. And after that, after we are adding and seeking for the vibration state, we can see the frequency and infrared also become different than the native state. And ER spectra also showed that there is a bonding at there, and the HOMO and LUMO pattern show the different pattern than the previous in native state. And here the electronic energy. In here, we can see that astaxanthin without metal ions show the lowest electronic energy, which means that it doesn't have to maintain or management electron in a better state than the astaxanthin with metal ions complex. And in entropy show that the equilibrium is far more likely easily to reach in astaxanthin with complex with metal ions. And also in head capacity, we can see the head capacity become more increased when we are adding astaxanthin with metal ions. And in binding site and interaction type, in this table, we can show that when we are adding astaxanthin with glycated human serum albumin, the binding site become minus 8.9 kilocalorie per mole. But when we are adding astaxanthin with CO2 plus and 2CO2 plus, it become more negative, which means it's far more likely to bound into the system more than the astaxanthin itself. And here we are comparing with metformin, as we know that metformin is the drug that we use for type 2 diabetes mellitus, and metformin, like the other drugs, also have side effects such as vomiting, nausea, and also the most disaster part is the lactic acidosis that could happen in type 2 diabetes mellitus. So we seek to the organic compound such as astaxanthin in this experiment. So when we are comparing we glycated human serum albumin with astaxanthin and glycated human serum albumin with astaxanthin complex with metal ions, there is still the same pattern with alkyl bond and hydrogen bond, but in the astaxanthin with CO2 plus and astaxanthin with 2CO2 plus, there are this interaction type, which is p alkyl bond happen in that stuff. And what does this result means? We can see in potential energy and binding energy, our data already achieve equilibrium, which means that the data is valid to analyze into the RMSD big bond. So we can see the stabilization that could happen in astaxanthin in glycated human serum albumin in pink. In the last part of 15,000 picosecond, we can see they are still going to be up higher with glycated human serum albumin. But when we are adding astaxanthin here in the blue, it become more lower than 3 nanometer. It means the stability of complex of protein already reached. But when we are adding astaxanthin with CO2 plus toward the glycated human serum albumin with one metal ions or two metal ions, it become more lower. Here it shows with black and red. So we can see the improvement when we are adding the metal ions toward the astaxanthin. Here in the last part, we can see the movie visualization. In A is astaxanthin in human serum albumin in human serum albumin protein in the 15,000 picosecond, or in the last of the molecular dynamic simulation, we can show that the site A and site B in human serum albumin still intact together. But when we are compare it into the glycated human serum albumin in B, we can show that there is the loss of unfolding even here between site A and site B. And when we are adding it with the astaxanthin, we can see in A, it become more intact and become more likely like the native one. And when we are adding the G and H here, it is for the astaxanthin with CO2 plus and astaxanthin with CO2 plus, it showed that it more become like the first one or the native one, which is the normal patient without type 2 diabetes mellitus or glycated human serum albumin. So the conclusion, we can show that energy values of astaxanthin plus CO2 plus and astaxanthin with 2CO2 plus are more negative than astaxanthin, indicating that they are more stable than astaxanthin in native form. And the second is astaxanthin metal complex has higher heat capacity, dipole moment, and entropy than ASX or astaxanthin. And this shows adding astaxanthin or astaxanthin metal complex to glycated human serum albumin increase protein stability and prevent from possible unfolding. And this result show that astaxanthin with metal complex better than astaxanthin in native form in order to inducing glycated human serum albumin to perform it is normal function. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh. Good afternoon.
Good afternoon. Thank you very much for your very interesting topic, Bapak Syahputra Wibowo. Now we jump to the last presenter for this session. It will be Ibu Tati Kusniati will deliver presentation about the characteristic of Lactobacillus Satsumensis EN38 alpha amylase and its potency in carbohydrate degradation in tuber pasifol. Ibu Tati, halo.
Okay, thank you very much.
You're welcome, Ibu. So time is yours, Ibu. You can do share your screen and
Okay.
start your presentation.
Yes. Is it all right?
Yeah, Ibu is already in my screen.
Oh, yes. Thank you.
You're welcome, Ibu.
Okay. Thank you very much uh, for moderator. And this afternoon, I would like to present our research with the topic of characteristic of Lactobacillus subsumensis EN3832 alpha amylase and its potency in carbohydrate degradation of sugar paste flour. The research background of this research, alpha amylase can be produced from within the human body and also from microorganisms, including lactic acid bacteria. Various alpha amylase producing lactic acid bacteria, including various species of Lactobacillus. Type of flour producing soup include cassava, sweet potato, and taro. Cassava, Manihot esculenta, can be used as a basic ingredient in the food industry as the main source of starch production. Sweet potato, Ipomea batatas, is a source of carbohydrate and a high source of calorie energy. And taro has been made into various processed products such as taro flour. Alpha amylase, which is widely used in the food industry, mostly come from import. To reduce import, local alpha amylase producing microorganisms need to be explored and their potential is revealed. Lactobacillus subsumensis EN3832 was an indigenous lactic acid bacteria which produced alpha amylase whose character and potential were not yet known in the processing of carbohydrate source food products. Therefore, the characteristic of alpha amylase produced by Lactobacillus subsumensis EN3832, its potency in the degradation of carbohydrate in local crops need to be studied. The results of this study are expected to be developed into carbohydrate food products that are easier to digest and safe for consumption, so that this food product can be consumed by both adults and seniors. The research method is material use, MLS growth media, DNS, Lactobacillus subsumensis EN3832 as indigenous bacteria, taro, sweet potato, and cassava. Subculture Lactobacillus subsumensis, W2 lactobacillus subsumensis, inoculate with MLS group, incubated 48 at 37. Preparation of lactic acid bacteria media for producing alpha amylase by using MNS fruit, and then sterilize for 15 minutes at 121 degrees Celsius, incubated at 37 degrees Celsius. Production of alpha amylase by using 2% L-sustumensis, and then sterilize at 121 degrees Celsius, 15 minutes, incubated 24 hours, 37 degrees Celsius, centrifuge at 9,000 rpm, 10 minutes at 4 degrees Celsius, and the supernatant alpha amylase take for food testing. Standard glucose curve, glucose solution at 500 of DNS solution, heat at 100 Celsius for 5 minutes at 1 milliliter distilled water for text measure and at sorbent at
wavelength 540 nm. And alpha amylase activity by using Benfeld method, one unit of alpha amylase activity was defined as the amount of enzyme whose reaction produced equivalent to 1 micromole of glucose per minute. And the alpha amylase activity, glucose concentration cross 1,000 cross deletion factor divided by glucose molecular weight cross 1 divided by time. Optimization of alpha amylase lactobacillus saxumensis use temperature from 30 up to 70 degrees Celsius. For pH from 3.5 up to 7.0. The highest activity based on the amylase activity test. The optimum of alpha amylase at certain temperature and pH. The stability of alpha amylase lactobacillus saxumensis, temperature from 30 up to 70, pH from 3.0 up to 7.0. The relative activity of alpha amylase above 5%. This expresses as stable activity. Degradation of carbohydrate in tuber pasta flour with the addition of lactobacillus saxumensis alpha amylase 1 gram tube pasta flour dissolved in 10 milliliter distilled water. Heat stir until 70 degrees Celsius and then add saxumensis and incubated at 37 degrees Celsius for 24 hours. Analysis of rejection sugar concentration by using DNS method and the formula rejection sugar concentration, glucose concentration ppm, divided sample weight, cross reaction total volume, cross dilution factor, cross 100%. And data analysis of optimization and stabilization of L-saxumensis alpha amylase analyzed by standard deviation and ANOVA, the data, standard deviation, and the data conducted in 3 applications. The research results show that the optimum activity of L-saxumensis alpha amylase was reached at a temperature of 45 and pH 5.5, and it will be shown in table 2. And the alpha amylase stability was reached at 30 up to 65 and pH 3.5 up to 7. It will be shown in table 3 up to 4. The rejection sugar of tub pasta flour in taro, sweet potato, and cassava with the addition of alpha amylase were 0.29, 0.0243, and 0.0244, while the rejection sugar increase were 2.333, 27.055, and 15.586%. And the rejection sugar of wheat pasta flour as a comparison was 0.030, while the rejection sugar increase was 54.442%. It will be shown in table 5. This one is table 1, the activity of L-saxumensis and 3832 alpha amylase in various temperatures. The temperature from 30 up to 70 and the activity from 0.1400 unit per milliliter, and the highest unit activity at 45 degrees Celsius as optimum activity. The value of 0.1889. And the table 2 activity L-saxumensis alpha amylase in various pH. The pH from 3.5 up to 7, and the highest at pH 5.5, the value of 0.3445. This one is the optimum activity of L-saxumensis alpha amylase. And the table relative activity of L-succumensis alpha amylase in various temperature and incubated for 1 hour. The temperature from 30 up to 70, and the activity for the optimum activity or stability 50% above activity. This one from 30 up to 65 degrees Celsius. The relative activity, the value more than 50 up to 100%. In this table 4, for relative activity of L-succumensis alpha amylase in various pH and incubation time for 1 hour. The pH from 3.5 up to pH 7, and the unit activity for the value 50% relative activity, and the value the above from 50% relative activity, all pH more than 50% relative activity. So from pH 3.5 up to 7, the L-succumensis alpha amylase was expressed as stable condition. For the table 5, increase in reduction sugar content of 3 local tub tester flour after addition of lactobacillus succumensis alpha amylase. The type of tub, taro, sweet potato, and cassava with as comparison, and with addition of alpha amylase from lactobacillus succumensis is tub, taro, sweet potato, and cassava. The reduction sugar content for taro plus alpha amylase 0.0291, and this value was higher than taro without alpha amylase 0.0285. For sweet potato plus alpha amylase, the value 0.0243, and the value was higher than sweet potato 0.177. For cassava plus alpha amylase, the value 0.0244. This was higher than cassava without alpha amylase 0.02, 0.6. For wheat as comparison, wheat plus alpha amylase, the value 0.03, 0.2. This was higher than wheat without alpha amylase 0.0137. For increased reduction sugar, the percentage of taro 2.3330. For sweet potato, the increased reduction sugar 27.0550, and for cassava, the increased reduction sugar 15.5860. For wheat as comparison, the increased reduction sugar 54.4420. For all the results, the increase of reduction sugar all tab from taro, sweet potato, and cassava, the 3 tabs were lower than increased reduction sugar from wheat as comparison. All the activity of alpha amylase were affected by type of species and media, and the stability of alpha amylase affected by type of species, temperature, and incubation time. The reduction sugar content in top pass floor affected by alpha amylase concentration, type of paste floor, and time of hydrolysis in top pastel floor. So the conclusion is the optimum activity and stability of L-succumensis alpha amylase was frequently reached at temperature of 45 with pH 5.5 and 30 up to 65 with pH 3.5 up to 7. The paste flour of taro, sweet potato, and cassava with the addition of L-succumensis alpha amylase had a lower reduction sugar content and reduced sugar increase compared to wheat pasta flour. show that the taro, sweet potato, and cassava fish flour with the addition of alpha amylase from Lactobacillus subcutaneous can be used with the same treatment as an alternative to wheat ester flour. Thank you very much.
Thank you very much for very detailed presentation, Ibu Tati Kusniati. Now we jump to Q&A session. So is there curious for our previous presenter? So I have curious for Ibu Linda, our third presenter, our second presenter in this session. Ibu Linda, may I ask you a question? Halo Ibu Linda.
Okay, sorry. Yes, please.
Okay, I saw that, and you have told that if some compounds from marine tunicates is effective to combat HIV virus, right? So I would like to know how effective this compound to combat HIV virus. Thank you very much.
Once more your question, I can hear.
Oh, all right, Ibu. So how effective the chemical compound that produced by marine tunicates in order to combat HIV virus?
Okay. I think definitely we have this alchemy from marine sources, and now, if I'm not mistaken, it's now in the two clinical trial phase. I forget of one or two, conducted by PharmaMar. I think we are curious to see how it works. I think it's now actually it's anti-cancer, but it's also now become the test for SARS-CoV-2. SARS-CoV-2.
All right. Thank you, Ibu Linda. So here is the end of our last session. So in the next session, Bapak Arif Lukman will take over my position here as a moderator. So before I move into the another room, so please apologize all of my mistake that I did. So thank you very much for your interest. Thank you and see you around.
Ladies and gentlemen, here please, I will introduce myself. My name is Arif Lukman. I will be the chairperson for this session, for the last session in this group.
Oh yeah, Bapak Arif Lukman, before I move into the another room, so we have to take a photo for this session.
Okay, sorry.
Okay, sorry. I forgot about it. So, Mbak Amel, you can take our picture. So please everyone turn on your camera.
Okay. Wait. Udah siap ya? 1, 2, 3. Okay, thank you.
Thank you, Mbak
Thank
Amel.
you. Terima
Thank
kasih.
you.
Thank you very much.
For the organizing committee, should we start right away or should we have a break for some minutes? Mbak Amelia, should we have a break for some minutes or just start right away for next session?
Maybe break 5 minutes, sir.
Yeah, okay, yeah, we will have a break for 5 minutes. Thank you.
Okay, thank you.